where we sit today, I said on the onset, molecular biologists are so intimately familiar with mRNA and your technology, which we'll get to in just a second. The reason why that is because, I get asked oftentimes by students, what do molecular biologists do day to day? And they do lots of different things, but a big proportion of them will spend their careers studying one, two, three, a couple of genes and asking every possible question about that gene. What does it do? What protein does it produce? What happens to the gene when it's to the protein when the gene's mutated? How does it affect the cell? Does the cell grow quicker? Does it die quicker? Does it look different? Does it behave differently? And so the reason why we're so intimately familiar is because we have to have a way in which to take that mRNA message, which is coding for some gene of interest, call it gene X, force it into cells and see what happens. And this is a process that we call transfection. And it is one of the most routinely done technologies in any molecular biology lab. So I want to get to that now. Can you please define for us transfections? And how did you find yourself developing this technology that is just indispensable? And personally, for me,
okay, so I'll give you the real story.
Give me the real story, and let's define transfection for our listeners.
And I came out of I got a biochemistry degree at UC Davis. There was not a molecular biology degree at the time, and I was working. I got my subsidy as a student came through working in a laboratory of the laboratory of Dr. Robert Cardiff and Murray Gardner at the Department of Pathology at UC Davis, working on breast cancer models. And I didn't realize at the time what the significance was. Bob Cardiff had just finished a sabbatical with Bishop and Varmus. So Bishop and Varmus got the Nobel Prize for the oncogene hypothesis. Mm-hmm. So he come out of that, and he was working with a model of breast cancer in mice involving a retrovirus. Now, the central dogma of biology is that DNA makes RNA, RNA makes protein. Retroviruses have a way to back that up, and they go from DNA form to RNA form, and that's what I think Temin and Baltimore got the Nobel Prize for. So retroviruses have this weird property of going back and forth. And one of the things they do is they insert themselves into the genome of mammalian cells. And when they do that, they disrupt the genes associated with wherever they've stuck themselves in. And they have their own molecular switches, promoters, enhancers. And so they not only affect their biology as DNA parasites, which is what they are, but they also affect the surrounding genetic information because promoters, enhancers, particularly enhancers, act at a distance. They don't just act proximately to whatever the genetic sequence is. So that was the world that I found myself in. And as I said, it was molecular biology as a PhD or a bachelor's degree didn't exist at UC Davis at the time. It really was just emerging. So I went through in my training basically the fundamentals of microbiology, and it was at UC Davis. You may recall the plasmids, the Puck series,
yes, of course.
Plasmid at the University of California, like Puck 19 and 18, PUC, et cetera, all those pairs, and also the single-stranded versions of those that were used in phage. That was all created at UC Davis in about the same time frame that I was there. So it was like one of these, like at the Salk Institute, one of these weird, happened to be at the right place at the right time where a lot of things were happening, including kind of the origins of dideoxynucleotide sequencing. And so because of the training I took, I was trained in the more classical microbiology involving, among other things, the use of a bacterial virus called a phage, particularly phage lambda, to insert genetic information and modify bacteria. Okay, so that is called transduction. The process by which a cancer cell becomes a regular cell becomes a cancer cell has a related nomenclature, transformation. Now, the irony is that, or the complexity here for the student, is that transformation is used in two different ways. There is a transforming principle for which the Nobel Prize was given that demonstrated that you could call, I think it was pneumococcus, that you could transform a pneumococcal bacterium from a rough form that was non-pathogenic to a smooth form that was pathogenic. And this goes back to World War II, as I recall, by applying DNA, is what it turns out to be, by applying a transforming principle that caused it to have this change. And so that was part of what gave rise to the awareness that DNA was this important information molecule, but it also gave rise to this nomenclature of transformation. And so it's used as transformation in a way is kind of synonymous with the word you just introduced, transfection. Transduction is the use of a virus, whether it's a bacterial virus or a retrovirus or recombinant retrovirus, like in gene therapy using a viral vector. Those are really transduction, and really transformation is the language that's often used there. And then there was this suite of technologies that were used back in the day for introducing intentionally, in a much more facile way, in a much more efficient way, small fragments of DNA into cells, whether they're bacterial cells or mammalian cells. And that came to be called transfection or infection. It's a derivative, right? It's you're infecting by transferring a material, but it's not really a virus that you're doing it with. So that was the nomenclature for transfection. And back when I was, so now we're getting to the story. Back in the 70s and 80s, this could be done through a couple of standard technologies. The leading one was by precipitating the DNA using calcium phosphate, so kind of the constituent of bone. And so you would make these really small precipitates, and there was an art in how small you could make the precipitate, how small you could make the crystals, because it seemed to work best if the crystals were really small. And then there was a whole art, it was kind of a little bit voodoo, of how you could treat cells, particularly bacterial cells, so that they would more readily take up this material, these small crystals. And that was called making them competent. And there's a whole art form around competent cells. You could buy competent cells from New England Biolabs. It was a whole thing. And you could look up in the Cold Spring Harbor manual, loosely bound manual of how to do stuff in molecular biology. I don't know if they still have it. They probably do. It's kind of like your field Bible about how to make competent cells for transformation. So that's calcium phosphate. And then there was another tech that involved the kind of clumping of DNA using a molecule called DEA dextran, small little beads. And that was another way that you could get DNA into cells. And then there was the hot new technology at the time in the late 80s, which was electroporation, using pulsed electrical fields to basically cause pores to form in the surface of cells, and particularly in bacterial cells. So that became kind of the method of choice in many ways, much more efficient than using competent cells in HB101 to deliver molecular information into a bacterial cell. And then eventually, and I was kind of at the forefront of that, that's something that's overlooked, is the development of the use of that tech of pulsed electrical fields in migrating from use for bacterial cells to use in mammalian cells. And then I really was absolutely the pioneer in then using that in vivo for RNA and DNA. That's never mentioned in all this chatter about the RNA world. So then now let's get down to the business. So I was busy trying to do reverse genetics, is another term, using RNA as a way to ask questions about a fundamental problem confronting the world of gene therapy, and particularly retroviral vectors. At the time, retroviral vectors, based on, I mentioned Murray Gardner a while ago, a virus that Murray had identified back in the day in mice, that had become the standard way for gene therapy, the idea that Ted Friedman of UC San Diego pioneered. This pediatrician came up with the idea, I think it's in the late 70s, that we could cure inborn errors of metabolism, ergo genetic diseases like muscular dystrophy, by transferring good genes into the bodies of children that had bad genes, that had inherited mutations. And so this very simplistic idea had been operationalized based on, remember I mentioned retroviruses that go from DNA into or RNA and DNA back and forth, and then they insert themselves into the genome of mammalian cells. And so retroviral vectors had been developed in where you cut out a lot of the genetic information at the center of the retrovirus and put in your gene of interest, maintain those elements of the retrovirus that are necessary for it to be transferred into the genome, and then complement the missing genes using an engineered mammalian cell that we call a packaging line. So that was the kind of the origins of gene therapy and the leading tech at the time when I entered Inder's lab at UC San Diego and the Salk Institute back in 87. So Inder was one of the top labs, and Ted Friedman was the other one, really, worldwide in the use of retroviral vectors. That's why I went there. And so now my story makes a little more sense. I come from mouse memory tumor virus, retroviral vectors, fundamentals of molecular biology, gene transfer, vaccines, which was a major focus in that laboratory at UC Davis. And I decided once I got into an MD PhD program that what I really wanted to do was become a pediatrician and cure inborn errors of metabolism, follow the vision of Ted Friedman. And here's a fun little aside. I went to Ted and asked him to be in his laboratory, and he said, no, I won't take you in my laboratory to work on gene therapy because it doesn't work.
Interesting.
Retroviral vectors aren't working. But if you want to come in and be involved in creating an ordered cosmid library of one of the key human chromosomes, I'd be glad to take you on for that. And I said, no. I've had enough sequencing for my entire lifetime. I don't want to do that for the rest of my life. And of course, what I did was I missed out on the opportunity to be an early player in the Human Genome Project. So that's the way careers go. But I wanted to do gene therapy. So Ted wouldn't take me, so I went to Inder. I was advised not to go to Inder. It was a wickedly vicious competitive postdoc lab of about 14 postdocs, and Inder was notoriously abusive. But, I was young, stupid, and ambitious. And so you go into a postdoc lab as the only graduate student, and historically, Inder would not take graduate students. And you got to figure out a niche. Otherwise, you're just going to get crushed. Yeah. This is the reality, for your students. Be careful what you wish for. So I went into this insanely competitive laboratory for molecular biology and virology, which had, at the time, I'm going to just give a little more anecdotes. Renato Del Becco was still there. His laboratory was across the hall from Inder's lab. Renato, of course, got the Nobel Prize for the fundamentals of mammalian cell culture. And this is the laboratory that had trained David Baltimore, had trained Susumu Tanagawa, other Nobel Prize winners. So insanely competitive, cutting edge environment.
Right place, right time for you. Amazing.
There was a postdoc, the leading gene therapy postdoc at the time was a guy named Dinko Valerio. Dinko was way out on the edge. He had abandoned retroviral vectors, and he was pursuing a whole new vector-based gene transfer technology that was right on the cutting edge, adenoviruses. Mm-hmm. And so Dinko was the pioneer, really, in many ways, in the same laboratory with adenoviruses for gene therapy. And when he left the laboratory, he created a company. This is another aside. The company was called Crucell. Crucell was later sold to J&J, and it is the Crucell technology that gave rise to the J&J SARS-CoV-2 vaccine. So that both of these vaccines that were marketed all came from Inder's lab originally. So here I am in this environment. I got to come up with something that is not going to just cause the surrounding postdocs to descend like vultures. And what I wanted to do was ask questions about how retroviruses got packaged, the RNA form of retroviruses, because we had some rudimentary information from reverse genetics and deletions about where the packaging sequence was for the various mouse-derived retroviruses. So this packaging sequence is at the 5' end, and it clearly we didn't fully understand how it worked. And so what I wanted to do was be able to create in vitro. And there was this hot new enzyme that had just come out, T7 RNA polymerase. Up until that time, everybody had been using SP6. And so there was kind of growing opportunities in RNA world. But all of that was still being used just as a way to produce proteins using a lysate of red blood cells, erythrocyte lysates. So you could take a reticulocyte lysate. This is a nucleated red blood cell, and you could make a protein preparation of it, and then you could add synthetic RNA to it, and it would turn that into protein. It had the ribosomes and the tRNA and the other components you needed. So ATP. That was kind of the state of the art right then. And I reasoned, what could possibly go wrong? We can make RNA in vitro, and we have packaging cell lines. That's why I told you the story about packaging cell lines that complement the missing factors that are necessary to assemble a retrovirus. We have packaging cell lines. We can make RNA in vitro. We can basically engineer that RNA to have whatever sequence characteristics we want. And then all we got to do is get the synthetic RNA into the cells. What could possibly go wrong? It took me a stupid amount of time to come up with a transcription vector that would produce a retroviral RNA, and also to start to ask the questions about, well, how can I identify a system for delivery, what you would call transfection, into a mammalian cell that would be sufficiently efficient that I could take these RNAs that I would make, put them into a packaging cell line, and get out a retrovirus, an infectious retrovirus. And this may sound crazy, but polio RNA is infectious. You can take purified polio RNA from a polio virus and put it on cultured cells, and you would get polio out. So the idea of RNA infectivity into cells and its use to produce an infectious virus, of course, it's infectious. That means it would be amplified in the cell culture. All you needed is a few polio viruses to originate from that, and then you could go and it would be amplified, and you could harvest them and all that good stuff. So what could possibly go wrong? Well, it turns out that DEA, dextran, and the precipitation using calcium phosphate with RNA is just insanely ineffective. It's just a fool's errand to try to get it to work. But I was banging my head against the wall trying to get this to work because the whole idea of reverse genetics was dependent on this concept. And this interesting guy who's still at the Salk, a true genius and a Nobel Prize also ran, named Tony Hunter, who keeps voluminous notes, was kind of a central hub of information in molecular biology at the time. Reviewer for Cell, all the top journals, etc. Worked at the other end of the hall from Inder. Inder and Tony often had kind of a frenemy relationship, let's say. And his other characters, Walter Eckert, it was an interesting time. So Tony one day comes to me and he says, "Hey, Robert, I'm recreating this is at the distance of decades, but Tony remembers that he still has notes about this, and he's not allowed to talk about it by the Salk. This relates to the patent issues that developed later on. But Tony came to me and he said, "Hey, Robert, there's this new tech Gordon Ringgold at Syntex has that you need to know about." Now, I think what really happened was that Tony reviewed Gordon's seminal paper on that Phil Feldner is on that involved the use of positively charged fats to deliver DNA. It came out of Syntex. Phil was working for Syntex at the time. And so Tony thought that it was Gordon because I think Gordon was the senior author. And so he said, "You ought to call up this guy, Gordon Ringgold, and find out about this new technology that they've got at Syntex and see if it works for your RNA." So I call up Gordon, and Gordon says, "Oh, I'm not the guy you need to talk to. You need to talk to this guy, Phil Feldner." Now, here's the story of that, the origin of the use of cationic lipids for transferring nucleic acids. Okay, this is the story that Phil told me decades ago. He never tells the story anymore. It's all Phil the genius. But this is another one of these strange kind of circumstantial events that often is at the root of breakthroughs. I mentioned that UC Davis was the source of the PUC plasmids and was very involved in the early days of transfection and expression of foreign proteins using bacterial plasmids in mammalian cells. And so a undergraduate student that had worked in that laboratory that had developed those vectors was doing, it's a woman, and I don't remember her name. I'm so sorry about that. It's like the Rosalind Franklin story, only a little bit smaller. So she was doing a industry internship at Syntex, which is kind of down the road in San Francisco Bay from UC Davis. And she found herself in the laboratory of Phil Feldner. Phil was assigned to developing ways to modify the charge on the surface of mammalian cells, which are generally negative. And Phil had come up with the use, Phil was a liposome specialist. There was some real core competence in the liposome world in the Bay Area, consequent to the development of the liposome company, the development of stealth liposomes in particular, their use for drug delivery, et cetera, et cetera. So Phil was part of that liposome world that had really developed to an advanced state within the Bay Area, Berkeley, UC San Francisco, et cetera, and Syntex. So Phil's job was, how can you take lipid technology and make it and adapt it for the purpose of modifying cell membrane surface charge? And so he took fusogenic lipids, known lipids that would insert themselves into the cytoplasmic membrane, that's the outside of the egg white in the metaphor, and modified them so that they would have a positive charge. In particular, he modified this using, now I'm going to get into chemistry a little bit, so follow me with this, stay patient. Quaternary amines are nitrogen-based molecules that have basically 4 carbon bonds, and it forces them into kind of a permanent positive charge. So quaternary amine is a permanent positive, as opposed to a tertiary amine. We'll come back to that because it's important, because it's tertiary amines that are used in the current formulations for the RNA in vivo delivery. And this is the work of the group up in University of British Columbia. So Phil was working with quaternary amine-based lipids, and this intern from Davis that had been messing about with transfection in the laboratories that had developed the Puck series, said to Phil, according to his story to me, when I asked him, how did this ever come about? She said to him, hey, DNA's negative. These things are positive. What happens when we mix them? Okay. And it was that simple. And that it is so fundamentally powerful, the combination of fusogenic lipids, self-assembling particles, in which you have this large, relatively speaking, polyanion, no negative charges all along the backbone of DNA molecule, and positively charged fats that will basically coat this. So it's not a liposome. That's a misnomer. Nanolipid particle is the right phrase. And it coats it, collapses it, creates a self-assembling particle. And this is key. A lot of folks like Judy White at UCSF, mentioned her name prominently because she played a key role in my work. And it's acknowledged in the first PNAS paper, postdoc in her lab. So Judy White was banging her head against the idea of, well, we could take RNA, messenger RNA, and encapsulate it in standard liposomes. So these bags of fat, and put it into the lumen of those bags of fat. That's a traditional liposome. And use that to deliver that RNA. So that was going on in parallel. I didn't even know about Judy back in the day. That came out later. So other people were trying to encapsulate RNA, but the problem is the entrapment efficiency is horrible. It's 1%, maybe 2%, because the concentration of RNA inside the bag is the same as the concentration of RNA in the solution outside the bag. And so what you do is things like sonication, where you basically are breaking these liposomes open so that you can get a gradient, so you can get the RNA to flow into the lumen. And then you got to purify them away from the other stuff, the solution. And making RNA is pretty expensive business, particularly back then. And so you ended up with this horrible entrapment efficiency, and then they wouldn't deliver for beans anyhow. So the delivery tech didn't work. And that was their frustration in Judy White's lab, and others at UCSF carried that on. So that was traditional liposomology. But what happened in the lightning struck at Syntax story is that this aha moment where this person came from a laboratory that was already doing DNA transfer and said, hey, this is negative, that's positive. What happens if we put them together? They self-assemble into a particle. And if you formulate that particle so that you have an excess of positive charge from those quaternary amines, remember what I said about cell membranes? Cell membranes are negative. So you end up with this basically ionically driven interaction of, it's kind of like magnets. Plus likes minus, minus likes plus, and they stick. And when they stick, if you have fusogenic lipids in the formulation, these are lipids that are known to disrupt membranes and have a tendency, if they're brought into close approximation, to flip from one membrane into another membrane. An example of that is diallyl phosphatidylethanolamin, which is why you have dope in many of these cationic lipid formulations. In addition to, I actually got the check mark for, I think, being the first to also add cholesterol to increase the fluidity of these membranes. That's one small paper in the, in the history. But if you use these fusogenic lipids together with these ones that have increased the fluidity of the lipid nanoplex, and it hits the cell, then you get these fusion events having to do with lipid transfer. And then that seems to facilitate the aggregate getting across the cytoplasmic membrane. Not really clear. The electron micrographs are hard to kind of freeze whatever that event is. And it appears that it is some strange hybrid of a fusion event and something akin to endocytosis. Because these particles can end up in endosomes.
I think it's important to mention that the membrane that we're trying to pass the genetic material through, it itself is also made of lipids.
Mm-hmm. Sorry, assume that to be fundamental knowledge. That white egg has membranes that separate the yellow nucleus from the white cytoplasm and separate the cytoplasm from the general extracellular milieu of fluids and everything else that's going on. And into that outer membrane is embedded a variety of proteins, and it is comprised of two layers. If my fingers are the hydrocarbon tails of these lipids that comprise these two, it's the membrane is a bilayer structure, and it has at its core, so it's what is the word? Amphipathic?
Amphipathic, yep.
At the core, it has a hydrocarbon, so it's oil-like. And at the outside, where my palm is charged molecules that are hydrophilic. They like water. And so you have water-liking molecules that also have water disliking or hydrocarbon components, and the hydrocarbon components associated with each other, and the charged components associated with water. And so they will form a lipid bilayer, which is what's used. It's a fundamental. We talked about the origins of biology and the role of RNA in the primordial molecule and all that. Well, without a doubt, one of the fundamental breakthroughs that may involve lightning or aliens from outer space or God only knows what, is the creation of an enablement of these small bags composed of lipid bilayers that allow the separation of one volume from the surrounding environment. And then that enables special magic to happen within the segregated area. So now we're getting into Brett Weinstein space. Yeah. So that's the one of the theories about the origins of life. But so you mentioned lipid bilayers, and I just had assumed that everybody understood.
Oh no, that's
that
okay. That's okay. But it's important because the biochemistry is related. So what did it feel like the first time this worked?
Okay, so what happens is, getting back to the story, Tony tells me to talk to Gordon. Gordon tells me to talk to Phil. Phil says, we have tried really hard to get RNA delivery to work, and we can't get it to work. In the interim, what had happened was that Ender sent me off to this laboratory at UCSF because, well, Robert, you can't get RNA delivery to work. You ought to go to a real laboratory that's doing this kind of work. And I know of one at UCSF, and it's the laboratory of Judy White. So I go up and I spend time in Judy White's lab, and I meet a postdoc and a Pablo, I forget his last name, I apologize, who had advanced the use of chloramphenicol acetyltransferase open reading frame. So CAT was the technology that everybody used for detecting gene expression back in the day. And it's a nasty business. It involves radioactivity and thin layer chromatography in order to create this biochemical reaction that you mentioned before, Western blots. So this is thin layer chromatography that would allow you to separate and show that a biochemical reaction had occurred, the acetylation of chloramphenicol via chloramphenicol acetyltransferase of a precursor radioactive molecule. So nasty business. You can't do many CAT assays. It's a big experiment to do a replicate of a 8-lane TLC plate. So that was the tech that was being used in Judy White's lab, was the production of a mRNA that encoded chloramphenicol acetyltransferase. And the thing that had made that go forward for them in that laboratory is that they had used a 5' and 3' untranslated region for the RNA derived from Xanthopus beta globin, as I recall, which is a very stable, highly translated RNA. And so what was done is to take those elements and put them into a cassette vector that would produce by SP6 a chloramphenicol acetyltransferase transcript flanked by the Xanthopus beta globin 5' and 3' untranslated regions that had a little short poly-A tail tag to it, because that's all you could get, because the larger poly-A tail is unstable in a plasmid. It self-deletes. So I got that plasmid from them. And then I had been, my first rotation at UC San Diego had been with this guy named Suresh Subramani. And Suresh had just published the paper in Molecular Biology, as I recall, involving his pioneering work on a whole new reporter gene called luciferase. And I had been in the laboratory. Luciferase is what made all this work. That's the honest truth. I would not have a career if it wasn't for luciferase, because basically it allowed you to now go and do at pennies and in minutes what it used to take hundreds of dollars and days in radioactivity and film exposure and blah, 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 blah with thin layer chromatography to do a CAT assay. So I took, I think Pablo is his name. I took his vector with the beta globin, pulled that out, dropped in luciferase, put it in a T7 context, and that became the workhorse. And I'd done that and demonstrated that I could produce luciferase RNA. And that had been the tech that I had used, the vector that I'd used with the DEA dextran and calcium phosphate experiments, which just gave me just the tiniest little signal over background. Luciferase is so insanely sensitive. It's like linear over 5 logs. And so you can detect the tiniest amount of luciferase, this is like femtogram levels in a luminometer, because it involves the production of photons that are very sensitively detected.
Right. So this is an enzyme that will produce a reddish glow of photons that you can detect.
Yeah, red green. It's the enzyme that makes it so that firefly tails glow.
Go. Perfect. And you were using this to show that you could get RNA into the cells that could then make this protein.
Because you had to have something that was sensitive enough that you could do the reporter gene assay. And so luciferase, so you had the combination. Luciferase is just so much more sensitive than chloramphenicol acetyltransferase. and so much more reproducible and linear over 5 logs.
Safe and cheap.
With cat, you would have to do the assay and you'd blow it out, and you'd have to redilute it and blah, 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 blah. Okay, luciferase, you could just do the assay, and you had this 5 log linear range, and what's not to like? So that was the platform, and then coming off of Judy White's lab, and then Phil said, we can't get RNA transfection to work here at Syntex. Maybe you can get it to work because you've done these other things. Well, the reason was because they hadn't done the heavy lifting of coming up with a reproducible system that was sensitive like this to be able to detect whether or not it was working.
Interesting.
So, Phil, at that point in time, I write an invention disclosure about the use of RNA as a drug, which was totally heretical. Remember, I'm in a retroviral vector laboratory, right, where the cutting edge is adenovirus vector. And I'm way out on the limb talking about the potential for using mRNA for gene therapy. Now, a reporter that I know from the New York Times, who's really buddy with Inder, another Indian, Purva is her name, talked to Inder about these days the other day. And he acknowledged that nobody was talking about doing this kind of stuff until I started doing it. So thanks, Inder. But that and $10 will get you a Starbucks. So that's what happened. Was I filed an invention disclosure. I talked to Phil about this. Phil said, we can't get it to work at Syntex. We'll send you some vials of this magic juice that he called lipofectin. That was this quaternary amine compound, DOTAP, together with and it's ether linked, so it's very stable. It won't degrade in cells, which is a bad thing. Together with diallyl phosphatidylethanolamine, which was a fusogenic lipid, so the positively charged lipid with a fusogenic lipid, formulated as liposomes, sort of, in a clear vial. So he sent that stuff to me, like 8 of them or something like that, with an NDA, and it was like grease lightning. It just put the two things together. This T7 RNA transcript with the Xenopus beta globin. And at that point, I was also doing there's another enzyme that will add a longer poly A tail, and I was using this new product that New England Biolabs was selling. They were called. No, it was Behringer. Behringer was manufacturing cap analog. So all that went into the tube, and then I did a little mini spin column to purify. So you end up with about $10,000 back in the day of enzymes and substrates in one reaction tube, which, I'm an unfunded graduate student. I don't know how I managed to get that through without Inder being aware of how much money I was consuming. But that's that. And then you'd have like a MIG of pure RNA, and you could do a bunch of experiments with it. So A plus B, put it on cells. Oh my God. The luminometer just goes.
And there you go. And the rest was history. And you mentioned earlier, your career would have not been what it was without luciferase. And there are many scientists sitting on this side who would say their careers wouldn't exist had it not been for the invention of transfections as you described them. So it's, for me, super fascinating to hear this.
So that's the actual story with all the warts and the serendipity and everything all added.
I'm glad you got a chance to get that out, because that needs to be heard. It's fascinating. So what that, just to put a bow on it, what that allows us to do now, sitting here in 2025, but even up 30 years ago, is we can now study gene function. You can take any gene using the technology that Dr. Malone just described, force it into cells, hijack the machinery of the cells to have them make the proteins, and away you go. For you've got a project that can last you 10 years simply because of this ability.
Yeah, you can do reverse genetics.
That's
I never got to solving the problem of packaging because this one little thing became so big. I mean, I can't tell you how crazy things went.
I could only imagine.
And then I was in an embryology lab, TAing an embryology lab, and I did what the hell? I have all these Xenopus embryos sitting around. And then we were doing the chick in the cup process for growing chick embryos. And I just, and this set off a firestorm eventually at UC Davis because I was doing unauthorized animal research. Of course, you don't need authorization to work with frogs with Xenophus laevis, but that was university politics. But suddenly I was with DNA expressing beta-galactosidase. I was producing Xenopus embryos that had taken up foreign genes, and so the folks that were involved in the embryology world people were, Robert, you have to collaborate with me. This is like the future of the world depends on it, and my next paper depends on it, and I could get funding for it. But it just, the greed. I ended up with a nervous breakdown.
Fair.
Which is why I ended up. I passed my qualifying exams. Focused on the issues of 5' 3' interaction, which was way out on the edge at the time in RNA world. And these various structural elements, the role of the 5' and 3' in translated regions, et cetera, in translation. And then all of this, there's a whole nother story that I've talked about in other places about the politics that went on. And I just got caught in the crossfire between University of California lawyers, Salk Institute lawyers, my mentor, quote, Andy Verma, other investigators at UC San Diego, and I couldn't take it anymore.
Yeah, academics is such a weird place. I mean, you've got such a fascinating story to tell in terms of creativity, human ingenuity, and then you've got greed, ego that comes in and stifles things.
The Bayh-Dole Act has, for all of its good intentions, has really distorted American science. So that was early on. I was trained in the Bayh-Dole. That's why I was filing invention disclosures. At the time, the Salk Institute under de Hoffman, who had been involved in the Manhattan Project, he was the leader. Inder took his place after he left or died. I think he died of AIDS. De Hoffman was trying to create Salk Institute Biologic Associates, which was their for-profit subsidiary of their not-profit institute. And so he blew a gasket when he heard about all this because Inder was collaborating with industry without getting authorization. It's just a bunch of ugly stuff went down. And I was, Dorothy, you're not in Kansas anymore. And all the things that you thought, I thought I was going to the ivory tower, the summit of academia at the Salk. And what I found out is it's just as petty and egregious as any other place.
Thank you for that story. That's absolutely fascinating for me. I hope others find it interesting as well. But for people who, like I said, use this technology day in and day out, it's kind of like, reading Genesis. And it's I thank you for that.